Ola Brynildsrud. I come from the Norwegian Institute of Public Health. And today I'm going to present you something that I worked with. And the name of the poster is Dancing with Phages. And it's about the dynamic interplay between Group A Streptococci and their viral partners, bacteriophages. So Group A Streptococci, normally it's associated with skin infections like impetigo and strep throat, but sometimes it can give these really nasty infections where it crosses into the bloodstream and gives stuff like scarlet fever or necrotizing fasciitis. And this can happen in young, healthy people, and it's not quite known why this sometimes happens. So in this work, I was interested in the virulence factors of Streptococcus pyogenes because it has a lot. In fact, it's more than 70 that we counted. So I tend to think of this bacterium as something like these 19th century battleships with just cannons everywhere. But sometimes these cannons can be removed. They're not always there. So the cannons are kind of retractable. That's where the phages come in. So Streptococcus pyogenes has something called superantigens. They're a very weird class of virulence factors because for all we know, they don't have any function except massively stimulating the immune system. So it provides a cytokine storm, and it just is very bad for the human host. And these virulence factors are typically associated with bacteriophages, but they don't provide any clear benefit to the phage itself, but they're very, very conserved. So why is this? That's not really clear. Does it provide an advantage to the virus? Does it provide an advantage to the Streptococcus? That's what I wanted to study. So in this material, I've studied 1,163 genomes from invasive Group A Streptococci. So these ones have all crossed into the bloodstream. These were whole genome sequenced as part of the reference laboratory program that we have at the Norwegian Institute of Public Health. And they've been sequenced with Illumina sequencing. So it's all short-read data, which is not ideal when you want to look at bacteriophages, because there's a lot of repetitive regions that those reads won't be able to completely resolve, but still we actually managed to resolve some of these phages. So this is a phylogenetic tree of all of my isolates. It's maybe not the most interesting figure on the poster, but it shows the different M types that we have circulating in Norway. It's mostly 1, 4, 12, 28, 87, and 89. And most of those are not associated with high levels of antibiotic resistance, but all of them are associated with different bacteriophages that can carry different virulence genes. So what did we find? I want to bring your attention to this figure, which is a phylogenetic tree of M1. So it's the most common M type in Norway. First, there's a clear distinction between what's known as the M1 UK type, which is a more virulent type of M1. That's on this red branch here. And well, first we wanted to know, is there a difference between M1 and M1 UK in terms of virulence genes? And we couldn't find any, even though we know M1 UK is typically associated with more, with worse courses of disease. But anyway, so we mapped all of these virulence genes and their presence or absence in all of these isolates. And some of the virulence genes are constant, right? They're in all of the isolates, and some are jumping in and out. Most of those jumping in and out are associated with phages. So here I've annotated all of the different genes that kind of jump in and out. So here, for example, we have a deletion of the SDA3, which is a DNase, which is associated with the phage. But maybe the interesting part here is there's a particular phage in the SF370.1 family. Well, it's not quite clear if it's in that family, but it does resemble the SF370.1 in its tail and integrase. And it seems to be kind of jumping in and out at a very rapid pace. And sometimes you would find isolates where the vertically inherited part of the genome is pretty much identical, but the presence or absence of this phage is different. So, for example, down here, we have 2 isolates that are carrying a version of this phage. And then here is another one which has just 2 SNPs of difference in the vertically inherited part of the genome, and the phage is not there. This phage carries a gene called SPEC. It's a superantigen, and it also carries SDA1, which is a DNase. So we were especially interested in this phage, and this was also one of the phages where we could fully resolve some of the variants. So we have described some new variants of this phage. Here is the SF370.1 at the top. And this is just a map of its genes. And here we have the different copies that we have found, which are variants of this phage, or by the definition, it's completely new genera because they are less than 70% identical by nucleotide identity. But as you see, they have kind of the same system here of tail fibers, lysine genes, and they carry these 2 virulence genes, the SPEC and SDA1 at the tail end. They're 100% conserved. They're identical. And also the integrase is the same between all of them. And you see there's kind of like a continuum here where you have these long ones that are 40,000 base pairs plus, and down to this very, very short one, which is just over 5,000 base pairs. So it seems to be some kind of continuum between these long temperate phages and this one, which must only have like satellite functions left. So variants of this phage was found in around 58% of our 1,163 isolates, and it seems to vary a lot between whether it's in a lysogenic or a lytic state. Jumps in and out, and I would like to explore whether this phage has anything to do with the invasiveness that can sometimes happen. And maybe it's time to rethink invasive Group A Streptococci. Maybe you can almost think of it as a viral disease. For this work, I'd like to thank my colleagues at the Norwegian Institute of Public Health that I have collaborated with.